In the last video, we were discussing about triglycerides. This time, we will be moving on to the next type or category of lipids, which is the phospholipid. So a phospholipid looks like this, right? It has a head and a tail. And at first, you might say that this is very different from a triglyceride. But if you really look at its structure, it's really just a modified triglyceride, if you think about it, because you still have the glycerol here, and then two are bonded with two fatty acids, and then the third bonding site is bonding with a phosphate group, right? So let me remind you how a triglyceride looks like before we move on, just so that you remember what I am talking about. So this is a triglyceride, right? So you have three fatty acids connected to a glycerol. And a phospholipid is again very similar. You have the glycerol, two fatty acids, and then the third bonding site, instead of another fatty acid, you have there a phosphate group. And this structure lends itself many advantages or uses because the head of this structure becomes hydrophilic or water loving, while the tail part, the two tails, the two fatty acids, would become hydrophobic. And this is because of the phosphate group here. So remember, oxygen is a very electronegative atom. So it hugs a lot of electrons, it tends to be negative. And if you look at oxygen here, this oxygen here has two bonds, this bond and that bond. This oxygen also has two bonds, but this oxygen lacking is lacking the second bond, so this tends to be negative. And even if it bonds with an oxygen here, the fact that this part here has three oxygens, it would tend to again be more attracting to electrons, so this would become partially negative. So the net effect is the head becomes polar, meaning it has charges. And remember, water is also a polar molecule because again of the fact that oxygen is a very electronegative atom. So you have oxygen here, and then you have the hydrogen here, right? If you remember your basic chemistry, because oxygen is very electronegative, what happens is this part of the molecule becomes partially negative, while this part becomes partially positive, right? Because the hydrogen is uh, not electronegative. So oxygens tend to become polar whenever it is part of a molecule or makes the molecule polar. It's the same thing here. So this head becomes polar, while the tail, if we notice, this is just a hydrocarbon. And remember, all the bonds here are kind of distributed, right? So this pull of the H here is kind of balanced with the pull of the H there. This pull of the carbon above is also balanced with the pull of the carbon below. So this overall becomes nonpolar, or it doesn't love water. In other words, this, if you can separate this is probably water soluble, and this is water insoluble, right? So this is a structure, therefore, that the head is different. It's hydrophilic, it loves water, but the tail is hydrophobic. It doesn't love water. And just to give you kind of a connection to your previous topics that you had in consumer chemistry, let me just remind you that the anatomy of a soap molecule is the same. Remember, a soap molecule is simply a long structure with a tail, where again the tail is nonpolar and the head is polar, meaning it has charges. And again, this is good because usually what we use for washing things is water, and usually the dirt that is in our skin, especially because of the sebum and our glands, usually they are oil based, right? And oil, remember, is non-polar. And the problem with that is water is polar, so water cannot dissolve oil. So therefore, if you wash oil with water, it will simply not take away the oil from your skin, so you will not be clean. And that's why people discovered to create substances like soap And this soap molecule is good because what it does is it will attract the oil via the tail, because the tail is nonpolar and the oil is nonpolar. But at the same time, the head is still attracted by the water. So think of it as the tail sticks to the oil, but the tail is connected to the head, and the head is attracted to the water. So when you wash, after you have applied soap into your skin, when you wash the foam, the water would wash away the head, but the head is connected to the tail, and the tail is now attracting the oil, so it will wash away even the oil in your skin. So again, I just want you to remember that, because that's kind of like an interesting fact, that you can a point of connection between consumer chemistry, soap, detergents, to this concept of phospholipids, right? But more importantly, in biology, phospholipids are very important, because guess what? They can create this structure. So if you remember, this is now a singular phospholipid here. Let me just annotate. This is a singular phospholipid here, right? And then another phospholipid here. And you create kind of like two layers, right? So this class is what we call a phospholipid layer, right? A phospholipid layer. And what is useful about that is this idea that when you have this structure, you can then kind of create a barrier. And in fact, that is what is used by the cell, right? So if you look at the bottom right of your screen, the cell membrane or the plasma membrane in all of your cells are made up of a phospholipid bilayer. And remember that this is important in biology, because cell is where life processes happens. And the thing is, it's important for the cell's environment inside the cell to be not affected by the environment, by the nuances, or by the factors outside of the cell. Because if there's no demarcation line between the environment and the internals of the cell, then if the environment outside changes, then it would also change the internal conditions of the cell, and the cell would not be able to function. And remember, life is a very delicate condition of specific temperature, specific pH, specific characteristics inside, so that the functions of the cell, like cellular respiration, can happen. Otherwise, it wouldn't work. So therefore, you need kind of like a barrier that separates the internals of the cell to the outside of the cell. And again, this is a good wall, so to speak, because see this, the heads here are water loving, the heads here are water loving, but the tails are water hating. So even if you have water outside of the cell, because remember when you have cells, this is one cell, another cell, another cell, another cell. In between those two cells, you still have some liquid or plasma kind of thing, or even blood in between, right? And therefore, there is water outside and water inside. But again, you need to kind of control what enters the cell and what exits the cell. And if you have this structure, then you prevent all types of things, because even if it's water soluble, when it gets to the center, it cannot just pass through immediately, because this is non-polar, right? If you have a steroidal compound, it cannot even penetrate the first layer, because the first layer is water loving, meaning as a consequence, it's oil hating. So it doesn't love steroidal based compounds or non-polar compounds. So that serves as a kind of like a gate. Obviously, there are things that can just pass through simple osmosis process, but the fact is, it serves as a barrier. And again, that barrier is good because it separates the outside environment from the inside environment, and therefore the cell can control its internal conditions so that it can perform life. So as you notice, phospholipids are very important because it makes up the plasma membrane of all your cells. So without them, we wouldn't be able to create cells. And cell is the basic unit of life, and therefore life as we know it wouldn't exist without them, right? So that is how important phospholipids are. And therefore, how this translates to your real life is that uh, you cannot just say, okay, I will be healthy, and they say that fats and oils, some of those, or we have this connotation that fat is bad, so you shouldn't eat fat. That is not also true, because these phospholipids are also based off of those things, right? So how you create phospholipids? You have, again, the fatty acids and phosphate groups, right? Where do you get the fatty acids? From the food that you eat, right? So if you eat fat based food like from animals, then those triglycerides, right, which you see here, those triglycerides will be broken down by your digestive system, and therefore when it's your turn to create phospholipids, right, when it's your turn to create phospholipids, then you would reuse those fatty acids. So therefore, it's not good to eliminate completely fats, right? And also there's a complication, because there are such things as healthy fats and not unhealthy fats. So but the thing is, we have to, again, science is so complicated. It's not a straightforward thing to say that this is bad for you, this is good for you. It depends on the use case. And again, from this perspective that all your cell membranes are from phospholipids, and phospholipids is based off of fatty acids, which you can get from fats and oils, then you should be consuming fats and oils. Obviously, everything should be in moderation. As my organic chemistry teacher in San Carlos used to say, everything is poison. What matters is the dose. So if you take a lot more than you need, then obviously it would become unhealthy for you. So there is a kind of balance between the amount of thing that you should intake. But this is the purpose of a phospholipid. It is very, very important in your cell, because again, it makes up the plasma membrane of your cell. So the linings of your cell, the even the covering of the nucleus, like the plasma membrane, that is made up of phospholipids. It's not just phospholipids, but the majority of that component are phospholipids. Okay, so that is about phospholipids. So now we move on to the next part, or to the next type of lipid, which is the steroid. So a steroid refers to this structure here. So again, this is a shorthand way of writing things, but as you know from grade 9, every vertex, all the vertices here are either carbon or oxygen. But this is kind of like a shorthand way of writing. So a steroid is just 4 fused strings, 3 of which are hexagonal in shape. Yeah. And 1 is pentagonal in shape, right? And what is that? Well, a steroid is usually based off of cholesterol, right? So cholesterol is this structure here, right? So a very complicated structure. But the thing is, the body uses that to produce all of these useful things. And these are just even the surface level. This is just 4 of the most common basic examples. So testosterone, estrogen, vitamin D, cortisone, those are based off of this, or even some parts of the plasma membrane, right? So again, as I told you, the plasma membrane is not just um, purely phospholipids. There are some components there, especially the gates that would allow steroids to come in and glucose to come in and other things, right? So if you look at this, here, testosterone, estrogen, what you would notice is that, sir, isn't that the same compound? Well, let's spot the difference. Oops, let's go back a little bit and let's pause on that frame. Let's spot the difference. So let's have here, this is O instead of an H, right? And all of the other structures are the same, right? So testosterone, therefore, is very much related to estrogen. And as you know, estrogen has a very different function than testosterone. So estrogen is the one that controls uh, part of the hormones that control the menstrual cycle. Testosterone is obviously for the male, so this is for sperm production and other secondary male features, right? But you would be surprised to know that the difference between the two is just this attachment here, right? So if we look at other steroid based compounds, let me remove these markings here. Like for example, well, cholesterol, we already know that. So now you have to understand what's the use case. And the use case is this. Instead of the body having to create from scratch, right? Instead of the body having to create from scratch estrogen or having to create from scratch testosterone, what it does is it just uses a, shall we say, a template structure. And that template structure is again this four fused rings, right? So think of it as, okay, if the body needs cholesterol, guess what? It will just take this structure and then attach these things, and that's cholesterol. And if it needs to synthesize estrogen, it will just simply remove this uh, connection here and then remove this and replace it with an O. And then this one, replace it with an OH. And now you have testosterone. But what if you are in a female body and you want to create estrogen? Then you remove this O here and attach H instead. So it's an efficiency thing, so that the body doesn't have to, let's say, reinvent the wheel every time. They will just take a template and then kind of like, okay, depending on the use case, okay, if we want estrogen now, we have to remove this, attach this, we have to remove this, attach that. If we want to create vitamin D, you have to remove this, attach that, remove this, attach that. And therefore, it makes the body more efficient. Now, in relation to what we call monomers and polymers, which is kind of like a running theme in this lipids or biomolecules in general. Well, the monomer there are the steroid based structure. Steroid, I will just call this a template. This is not a correct scientific term, but what I mean are the four fused rings. So you have hexagon, hexagon, and then another hexagon. And then here you have a pentagonal shape. That is the basis, the monomers. The polymers that they can create are all steroid based, like cholesterol, testosterone, estrogen, vitamin D, cortisone, all those other stuff. Now, do you need to remember what those are? Nope. Because again, we are not into biochemical pathways. If you, when you take up when you go to medical school, you would sure memorize them. But for high school level, we don't need that. We just need to know that all of those, estrogen, testosterone, cholesterol, vitamin D, cortisone, and others, many others, they are based off of the template, and the template is uh, the steroid four fused rings structure, right? And again, technically speaking, it's not a monomer polymer thing, because monomer tends to suggest that you have basic units, and then those basic units you kind of connect and connect and connect and connect and string them together, like amino acids create proteins, like nucleotides create RNA, DNA, like the monosaccharides create the polysaccharides and so on. But in the sense of building something more complicated from a simple template, then this is its equivalent, that we based it off these 4 fused rings. Okay, so again, that is all about the steroids. Now we go into the next one, which are waxes. And waxes are kind of different in terms of structure because waxes have no specific structure. What I mean by that is some waxes could be triglyceride in form. Some of them might have some phospholipid components. Some of them might have steroid based components. Some of them are simple hydrocarbons, meaning alkanes or alkenes or alkynes. And remember, all of those are insoluble, right? Yes, I know in phospholipids, the head is water loving, so it's technically um, can be dissolved, but not the whole molecule, just that part. So as a whole, the phospholipid is still not soluble in water. It's just the head part that attracts water. But as long as it's insoluble, it could be triglyceride in form, phospholipid in form, steroid in form, or even just a nonpolar hydrocarbon like alkene, alkyne, and so on. As long as it's insoluble, and waxes are defined by being used by organisms externally, right? Externally. So we can't find these waxes inside the bodies of organisms. So let me repeat. Number one, there's no specific structure for waxes. It could be triglyceride in form, phospholipid in form, steroidal in form, or just plain hydrocarbons like alkanes, alkynes, and alkenes. Number two, they are solid in form at room temperature, right? So solid in form at room temperature. And number three, they should be found outside of the organism's body. So for example, the leaves, the wax coatings in leaves of certain plants, it's a steroidal form of wax, and it's placed there obviously to protect. It's not that it doesn't want water. The leaves is not coated with wax because it does not want to be wet. Actually, it's the opposite. The wax is actually there not because it doesn't want water to enter. It's the opposite. The wax is there so that water that is already in the leaves cannot escape. Okay? Because you see, plants do not really absorb water through leaves. They absorb water through the roots, remember? And therefore, when they have water already, they send it to the leaves for photosynthesis. The bad thing is with photosynthesis, it needs sunlight. And you see, water plus sun energy, it will boil off that water. And the plants do not want that. So they have like a wax coating so that the water that they have just gotten from the roots and forwarded to the leaves will not escape. So that's the use case for the wax. Obviously, you have other examples of waxes, like the ear wax. And again, technically speaking, that's still in external. It's not, technically inside, because that the earwax resides, on the outside part of the ear or the ear canal. It's not really inside, right? So earwax, for example, that's also a kind of wax. Beeswax. So therefore, those are the types of so let me repeat, because I am not sure if I was showing you the screen. But again, waxes are insoluble in water. They are solid in form. They have no specific structure, and they are found outside of the bodies of organisms. So you can have wax coatings in leaves, earwax. You can have beeswax. Those are the types of waxes. So let me repeat. For a wax to be a wax, it has to comply those requirements. Number one, it must be insoluble in water. Well, that's a given because lipids are generally insoluble in water. Number two, it must be solid at room temperature, right? Solid at room temperature. Number three, it has no specific structure. It could be triglyceride in form, phospholipid in form, steroids in form, or just a simple alkane. And number four, they are used outside of the bodies of organisms. So again, typical example would be the waxes in the leaves of plants, earwax, beeswax, and all other types of waxes. But that concludes all the categories of lipids. So again, in the previous video, we discussed triglyceride. And then in this video, we discuss about phospholipids, how it's related to triglycerides, steroids, how it may not have the typical monomer polymer structure, but again, you can think of the main um, steroid structure as the four fused rings as kind of like the basis for making complicated compounds. So in that sense, that is the monomer, right? But yes, the monomer polymer structure here is loosely applied to this kind of template, kind of situation in this specific example. Now the waxes, it has no monomer polymer structure unless of course it's triglyceride in form. If it's in triglyceride in form, then the structure is just simply the same, glycerol and 3 fatty acids. For phospholipids, it's glycerol, fatty acids plus a phosphate group. For steroids, it's the 4 fused rings. But again, for waxes, there's no specific structure for that. So that concludes our explanations for the biomolecules of lipids. So again, 4 types of biomolecules: triglyceride, discussed in the previous video, and then phospholipids, steroids, and waxes.